the word DNA. Yes, it is actually genetic material in our body which transfers information from one generation to other generation. Similarly, there is another molecule, RNA. Actually, this DNA and RNA are nucleic acids. They are made up of combination of ribose, sugar with a nitrogenous base and a phosphate group, and they are present in the form of long polymers actually. So let's start their study. Nucleic acids. Nucleic acids are basically polymers of nucleotides, and hence they are known as polynucleotides. We'll study what are nucleotides in just coming slides. And these are of basically two types. Deoxyribonucleic acids, you must be aware of DNA. It is deoxyribonucleic acid. Another is ribonucleic acid, which is known as RNA. These two are genetic materials in fact. And each nucleic acid consists of a pentose sugar, a phosphate moiety, and a nitrogenous base. And basically that pentose sugar is ribose most of the time. In case of DNA also, and in case of RNA also, it is deoxyribose. This is ribo sugar. So the pentose sugar is ribose. Apart from pentose sugar, there would be a phosphate group and there would be a nitrogenous base, which is a heterocyclic compound containing nitrogen. We'll study about these also. So this is overall, nucleic acids are the polymer of nucleotides. That's why they are known as polynucleotides. Nucleic acids are of two types: deoxyribonucleic acid DNA and ribonucleic acid RNA. And these two are the genetic materials actually. And each nucleic acid, which is made up of nucleotides, nucleic acid is a chain of nucleotides, and that nucleotide contains a phosphate group, a pentose sugar, and a nitrogenous base. And this is the actually structure. This is the way they are attached. A 5-carbon sugar, generally ribose. On this side there is a phosphate group, and in this side there is a heterocyclic compound containing nitrogen known as nitrogenous base. So this is an overall structure of a nucleotide, and these form long chain to form nucleic acids. So the first thing is nucleoside. When there is a pentose sugar and there is a base, 5 sugar, then this combination is known as nucleoside. Nucleoside is ribose sugar or a pentose sugar with a heterocyclic compound containing nitrogen, that is nitrogenous base. So a nitrogenous base combines with a pentose sugar to form nucleoside. Now when there is a phosphate group also present, it would be known as a nucleotide. So combination of sugar and base, nucleoside, and sugar, base, and phosphate group, nucleotide. And these nucleotide combined together, two nucleotides combined together to form a dinucleotide, and the addition takes place continuously to form a long chain, which is known as polynucleotide or nucleic acids. So let's say this is a nucleotide with a sugar, a base, and a phosphate group. Similarly, this is another one, and these two combined together through phosphate linkage, which is bond between this phosphate group and this OH group. These combine together to form, this is called phosphodiester linkage. So far in this chapter we have studied about 3 linkage. First was glycosidic linkage in case of carbohydrates. Second was this is phosphate linkage, and there was a peptide linkage in case of proteins. So pentose sugar, nitrogenous base, nucleoside, pentose sugar, nitrogenous base, phosphate group, nucleotide. Two nucleotides or more nucleotides combining together through phosphodiester linkage, polynucleotide or nucleic acid. And we know there are two types of nucleic acid: deoxyribonucleic acid DNA and ribonucleic acid RNA. So what is the difference between these two? That in DNA there is a beta D2 deoxyribose. So this is beta D ribose, and this is beta D 2-deoxyribo. In case of RNA it is beta D ribose. In case of DNA it is beta D 2-deoxyribo. So this is ribose actually, and this is actually 2-deoxyribose. This is the way they are. So what is the difference between these two? This is carbon number 1, this is carbon number 2. This is a typical ribose. When there is only H group in place of OH group, it becomes 2-deoxyribose. That means an oxygen is missing. So this 2-deoxyribose combines with a phosphate group and pentose sugar to form DNA, and this ribose combines with base, nitrogenous base and phosphate group to form RNA. So this is the only difference between DNA and RNA, that in DNA there is a deoxyribose sugar, while in case of RNA there is a ribose sugar, simple ribose sugar. So now we have talked about nitrogenous bases. Now what are these nitrogenous bases? In case of DNA, the bases are either adenine, guanine, cytosine or thymine. In case of RNA, the bases are adenine, guanine, cytosine and uracil. The difference is between this, thymine and uracil. All the bases are same in case of DNA and RNA, but the difference is that in DNA there is thymine, in case of RNA it is uracil. And this is the structure actually. DNA is a single strand structure, RNA has single strand structure, while in case of DNA there are two strands with a helical structure, and these two strands are held together through hydrogen bonding and bisulfide bonding between the bases. This is deoxyribonucleic acid DNA, this is ribonucleic acid RNA. And this is the structure of bases. This is adenine, this is guanine, this is cytosine, this is thymine, and this is uracil. So these nitrogenous bases combine with pentose sugar and phosphate group to form these nucleotides, which combine further together to form long chain of polynucleotides known as nucleic acids. Now the structure of DNA and RNA actually. In secondary structure, the helix of DNA are double strands, as we have studied. These are double strand structure, while those of RNA, these are single strand structures. Two strands of DNA are complementary to each other. This is because hydrogen bonds are formed between specific pairs of bases. I've told you that DNA is a double strand structure and it becomes helical in structure because of hydrogen bonding formed between the base pairs. But the hydrogen bonding is formed between specific pairs of bases, and that is why there is a specific pattern of the two strands, and those are complementary to each other. And this is the way the bond is formed actually. AGTC. That means adenine forms hydrogen bond with always guanine, and thymine forms hydrogen bond only with cytosine. So this is the base pair actually. This is RNA single stranded, this is DNA double stranded. And now the difference between RNA and DNA. RNA is not responsible for heredity. They are actually messenger. They carry messages from one place to another place within the neural system, while DNA is chemical basis of heredity. They transfer the genetic information from one generation to other generation. While proteins are synthesized by RNA molecule within the cell, but DNA molecules do not synthesize proteins but transfer the coded messages from synthesis of protein to the cell. So what is the basic difference between that? That RNA is not genetic material generally, DNA is genetic material. RNA is genetic material in some bacteria and some plants, but in animals RNA is not genetic material, DNA is genetic material. RNA synthesizes protein, DNA does not synthesize protein. Apart from that, RNA are of different type, like messenger RNA, synthesis RNA, so there are different works of RNA, but DNA is not involved in any kind of synthesis. It only transfers information from one generation to another generation, from one cell to another cell. Students, in case you have any doubt in this topic, you can post that question in our discussion board. For that, all you need to do is to go to the home page of Ask IITians. There you will find an option of discussion board. You have to click there. Then in the right side, you will find a portion of Ask Experts. In that, just select the category in which you want to post your question. Let's say it's physical chemistry. Then post your question here, and then if you want to attach any image, use this option to attach the image, and finally post it by clicking on Get Answer, and you will be answered within 24 to 48 hours by our experts. So students, we studied about nucleic acids also. With this chapter finishes, and in this chapter we started with carbohydrate. We studied about monosaccharides, disaccharides, polysaccharides. Then we moved to amino acids, essential amino acids, non-essential amino acids, their properties, their structure. And then we moved to proteins and also nucleic acids. Actually, this is a very easy chapter. It has very little weightage in IIT-JEE also, but the questions which are asked from this chapter are actually very easy. So you can score those marks very easily if you study this chapter with little bit of careful. Okay, so this chapter is also important for exam point of view for IIT-JEE and also for medical exams. For more videos on physics, chemistry, biology and mathematics, stay tuned with Ask IITians. Thank you.